me? Can you hear me? Yeah, it's better, so try to adjust. Okay, like that, it's okay, more or less, like that. Okay, so good morning, everyone. First of all, I would like to thank the organizers of this workshop. Staff matter is an interdisciplinary field, and yesterday we mainly focused on theoretical physics. So now we are going to shift to material engineering. In my research group, we use hydrogels as soft matter, and we combine it with an emerging technology, which is 4D printing. So I am Mathilde Champeau. I'm assistant professor at the Federal University of ABC, which is located at 40 minutes far from here, in the southeast of São Paulo. ABC region is an industrial region, and we have two campuses, one in Santo André and one in São Bernardo, and our university is quite young. We have undergraduate programs in physics, chemistry, biology, material engineering, biomedical engineering, and much more. And focusing on materials, we have a master program in material science and engineering, and a master and PhD program on nanosciences and advanced materials. So if there are some students interested in doing a master or a PhD in this field, please come to talk to me during the coffee break so I can give more information. Applications are opened. Even if it's a young university, we are well equipped, particularly with characterization technique. We have a multi-user experimental facilities that give to the research community access for free to a large range of material characterization. And as you will know tomorrow, we are very close to the St. Payne or Sirius with the E. And so we are users of these facilities, as I will show you. 5 years ago, we get financial support from the CNPq. It was a low amount of money, but sufficient to perform all the work I will show you. We succeeded to buy this very simple 3D printer, extrusion-based 3D printer, and my student also built this one. And so, now, talking about the scientific aspect of this talk. So first of all, just to remember what is a hydrogel. So a hydrogel is a three-dimensional network that is able to absorb a large amount of water and that doesn't dissolve. So to avoid this problem of dissolving, we use chemical or physical crosslinks between the macromolecules. So we have natural hydrogels such as collagen, gelatin, and a large range of synthetic hydrogels that can be used and synthesized. So just to tell you, hydrogel synthesis is not such complicated. So basically, we choose a monomer, which can be liquid or dissolved in water, and we transform it in large macromolecules, and we use crosslinker, in that case chemical agent, in order to crosslink this long polymeric chain. And to do this polymerization and crosslinking, in that case we use energy, that can be UV light or temperature. In our case we are using UV. So here I showed the polymerization of polyacrylic acid that I will use in the project I will show you. And 3D and 4D printing. So I'm sure all of you know what is 3D printing, and I'm not sure you know what is 4D printing. So just to remember, 3D printing is an additive manufacturing process. We construct layer by layer our structure, and the objective of using 3D printing is the precision that we get in the shape of our structure, so we can build complex structures. So basically, we place what we call an ink in this syringe. We apply the pressure on it, so the gel is extruded and deposited on the platform. And there are many parameters we need to adjust in order to control the quality of the filament that is deposited, such as the syringe velocity, the pressure, the needle platform distance, the diameter of the needle, and so on. So it's not so easy as it may appear. And in 3D printing, we get static object. So this is a demonstration of here, constructed with hydrogels. So first of all, from a medical image, we reconstruct a 3D CAD model. After that, it is sliced with a program. Then it is sent to the 3D printer that will build layer by layer this structure. Then we remove the support material that is eventually used, and we get our static object. And here is just to show you the precision that we can get. So we can get a filament with micrometer sized. But what about 4D printing? In 4D printing, we print a 3D object that responds to time. So the fourth dimension is time. Our structure is able to respond to a stimulus that can be light, temperature, pH, ionic strength, and so on. And it entails a shape morphing or a change in properties. And to my point of view, it depends on the definition, but we have a 4D printing when the structure can reversibly respond to the stimulus. If we stop the stimulus, it comes back to the initial shape, and so on, during many cycles. So here are some examples. The first work has been published a few years ago. It is a flower that is thermoresponsive, so it has been built with a thermoresponsive polymer. Here we have two material structure. The red one is thermoresponsive. At low temperature, it swell. At high temperature, it shrink. And so we get this movement for this tube. Here it is a grip that is pH sensitive. So at high pH, it swell. No, it shrinks, sorry, so it is able to grip this flask. Then it is placed in a tank with a low pH, and so it's open again, and so it's like an actuator. And in that case, this small octopus has been obtained by placing ferromagnetic particles, nanoparticles, in the mixture, and by applying an external magnetic field, so we can make this octopus moving. But to get this four dimension, we need a swelling mismatch between two regions of the printed object. So this creates a stress, and so the structure is able to change its shape. So this swelling mismatch can be obtained in just one filament. You expose it to UV, and you will create cross-linking gradients. So on the top, it will be more cross-linked than in the bottom. So then you place it in water under a stimuli, and the upper part will swell less than the bottom part, and you have such kind of movement. Either you can also work on different layer orientation, and so depending on the orientation of the two layers, you will get different movements. So to get program movement, you obtain program movement by controlling the local material composition and cross-linking density, as well as the spatial arrangement of your materials. And so 3 years ago, we published this review in order to understand what has been done, which kind of the material have been used, and so on. And we end up with two categories of hydrogel. Stimuli-responsive hydrogel have been used, and non-responsive hydrogel can also be used. So from the first category, we have some hydrogels that respond to temperature, to pH, ionic strength, electric field. So this is the case here. We have this small robot with a thin arm. It is placed in water. Two electrodes are placed apart. An electric field is applied. And in this thin filament, we have a charge movement that occur. Osmotic pressure is created. It swells more on this left part, and so we have the bending movement of this arm towards the anode. And so it can be used to grip this little ball, and if we wanted to release this ball, we invert here the electric field, and so the ball is released. We also have a light that can be used, and magnetic field, which was the case of the octopus. In the case of non-responsive hydrogels, we can bring with the cross-linking density. So in this work, between these two glass slides, we have ink that is photo curable. We apply a control light with a control intensity. We end up with different cross-linking degrees here, the same on the bottom. We take this hydrogel and we remove the unreacted parts, and then drying it and placing it in different solvents, so we have this movement. So this is the printed hydrogel. When it is desolvated, we have this origami, and then it comes back to its original shape. Okay, so what are the applications of 4D printing? Some applications emerge in the field of sensors, such as valves that are sensitive to the temperature of the fluid. Here at 20 degrees C, you see the fluid passing right here can flow. At 60 degrees C, it is stopped. Actuators, so I have already shown this application in architecture. So it is just an idea from those that have been proposed by the Institute for Advanced Architectures of Catalonia. They have proposed to build a roof containing hydrogels. When the outer temperature is high, the hydrogels shrink, so it releases water. It releases water inside the building, and it was shown that it is able to decrease by 5 degrees the internal temperature of the museum. So talking about global warming, this is definitely something that as engineers, we wouldn't think, but that is really smart, and it can be produced by 4D printing. And in tissue engineering and drug delivery, we can expect nice advancements. But what about our work? Our approach is to work with these techniques, these 3D printing techniques, which is extrusion-based techniques. Many technologies exist, but we focus on that one because it is the most widespread technique among the laboratory. So if you go to talk to biologists, doctors, and so on, all of them will have this kind of technique in mind and in their laboratory. But the problem is that to reach such application and so on, we need to have kind of a high range of possible hydrogels that can be used depending on the application, which is not the case. So our focus is to develop new material composition, but also we want to furnish like a recipe to these people. So we want to optimize rheology, but also give which printing parameters should be used in order to have a nice filament and precise structure printed. So we just work currently with very simple programmable structure just as a proof of concept. And so the two projects I'm going to present are this one. The first one is to have a pH-sensitive structure, and the other one a temperature-sensitive structure. So in the first one, we use a combination, a mixture of polyacrylic acid, the guy that I shown the synthesis, and the pluronic. So pluronic is a thermal-sensitive polymer that is widely used. It is the gold standard in bioprinting. At very low temperature, it is miscible in water. It forms this solution. And when we increase the temperature, we get micelles that are formed. And increasing even more the temperature, we have liquid crystal that is created. And so it jellifies. So we can place the gel in the syringe and then control the flow rate of pluronic. But our goal is to develop a pH-sensitive structure. So we've selected a polyacrylic acid that is pH-sensitive. Here you can see this polymer at low pH that shrink. At high pH, it swells. And just for the history, it is a polymer that is used in diapers, baby diapers. So for those who have babies, this hydrogel is saving our lives. And so what we do, we mix pluronic and acrylic acid, the monomer. Then we expect it to gellify when we increase the temperature. Then we print it, and once the structure is printed, we apply UV light, so we get a polyacrylic acid. But the question is, how much acrylic acid should we mix with pluronic and keep the gelation properties and adequate rheological properties. So we fix the concentration of pluronic and we change the concentration of acrylic acid. And the first test that we perform is very simple, is a test of inverse tube. So we place a small volume of this different composition in a tube, we place it in a thermostated bath, we increase step by step the temperature of this bath, and at each temperature, we take it, reverse it, and if it's flow, it didn't gellify. If it it doesn't flow, it jellifies. So we determine the gelation temperature. For pluronic, it is 17 degrees C. For 16% of acrylic acid, it is 9 degrees C. Then it increases again, and in the case of 20% of acrylic acid, it doesn't gellify, so it is not adequate for 3D printing, otherwise it will drop from the syringe. And then, from a fundamental point of view, we wanted to know if acrylic acid can impact the liquid crystal formed by pluronic micelles. So we went to Campinas at the same pane that you will visit tomorrow, and we performed SAX measurements. So it's nice because yesterday Anton Parr has already explained how SAX works, so I don't need to do. Basically, we get this information as a scattering vector as a function of intensity. We look at the peak position right here and on the ratio between this first peak and the other one. And depending on this ratio, we can determine which is the liquid structure that is formed. So in case of pluronic, we end up with the FCC structure. And when we mix it with acrylic acid, we end up with a BCC structure. So there is an influence of acrylic acid on the liquid crystal that is formed. And now we are trying to understand, to explain why, due to hydrogen bondings between the different parts of this copolymer. So of course we need to look at rheology. So first, measurement is the measurement of the viscosity as a function of the shear rate. In all cases, we get a shear thinning behavior, which is adequate for printing. We need this decrease of viscosity when we apply a shear rate. And then we have a look to the amplitude sweep test, which measures the storage modulus and the loss modulus. And we want to have the storage modulus, which are the square, higher than the loss modulus, which are the stars. So it is the case in pure pluronic, in the one with 16% of acrylic acid, but it's not the case with 18% of acrylic acid. So it seems that the 16% of acrylic acid is more adequate for printing. So in that case, what does it mean? It means that we will have shape fidelity. So when the squares are higher than the stars, we have such kind of quality, shape fidelity, whereas in the other case, we have such kind of behavior. So it's the filament will spread in the platform. Very quickly, another test that we perform is we try to mimic the shear rate that the ink will experiment during printing. So at the beginning, it is at rest, so very low shear rate. Then it experiments a large shear rate, and then low shear rate again. And so we look at this viscosity profile. We observe that we have no exotropy. Here there is no change of viscosity with time. And we also have a look to this time required to reach this viscosity. And so we see that in the case of the 18%, the time to reach this high viscosity is 14 seconds, so it can impact the shape fidelity. So other optimizations that we do, I won't have time to show everything, but is the UV exposure of the printed structure in order to get the desired cross-linking density. Also, we check the stability of the hydrogels in water at different pH and temperatures, since we used pH-sensitive material and temperature-sensitive. We optimized all the printing parameters, and here is what we... I wanted to show you a video. Oh, I think I made a mistake. Okay, I will show you here what we get. So here is a filament at high pH. Placing in low pH, it shrinks. No, sorry, in high pH. So the outer part swells, so it entails the buckling of this filament and coming back to this low pH, so it opens again. So it takes a little bit of time, so this can be a problem for application, but we check the shape morphing. So I think it is not sharing anymore my presentation. Can I continue? Okay. So sorry for those who are assisting online. So then in the second project, we work with temperature responsive polymer, which is very known in the field of hydrogel, which is PNIPAM. So PNIPAM has a low critical solution temperature, about 32 degrees C. At low temperature, it is hydrated, swollen, and at higher temperature than 32, it dehydrates, so it shrinks. It is very fragile, so we need to reinforce it using this other polymer, so we mixed both of them. And in this project, we wanted to use an anisotropic nanoparticle, which is cellulose nanocrystals. We mixed all of them, then we placed it in the syringe, we print it, and when, so here in this mixture, the CNC are randomly orientated, and when it experiences shear stress here, so this little rod will align with the shear stress. And so in the filament, we expect to have an orientation of this CNC along the direction of the filament. And due to this anisotropy in the filament, we have different swelling in the two dimensions, the two directions, and this ends up with this shape morphing. So we had a lot of difficulty to disperse the CNC in our ink. So here it's an optical image, and we see aggregates of CNC. We tried a lot, and we didn't succeed to make them disappear. So we said, let's try to print it and see what happens. So we printed it, and then we checked our filament by atomic force microscopy. We see that we have some region, so here on the screen it's not so good, but we have some region where we have alignment of CNC aggregates in some region with individual CNC that are aligned with the shear stress. So after that, we optimize the printing parameters. We check the shape fidelity for all this composition, and we saw that the 10% CNC is the best. We confirm that we have temperature responsiveness, so at low temperature we have this extended filament, and at high temperature very quickly this filament shrink and fold. And we check that depending on the angle between two layers deposited, we can have twisting of our printed shapes. So as a conclusion, this is a very recent research field. I didn't mention, but for the first time, 4D printing has been mentioned by Tibbits, which is a professor from the MIT, so 10 years ago. And so it is on, there is a lot of effort that are being done in the development of new material and functional program structures. And in the literature, we see some fanciful applications, but I guess that very real application, relevant application will come. But since we have to deal with a good material for the application, a good material for being printed, the whole project is very complex. So I guess that in few years we will be able to see some very relevant applications. And so I would like to thank all of you. I would like also to thank all my students working in this project. Here it's a picture where we went to the former synchrotron. We haven't experienced already the use of the new synchrotron. And so thank you to all of you for your attention.
I have a quick question. We had lots of talks yesterday about active matter, for instance. Can you 4D print a swimmer, for instance, a self-propelled swimmer?
Yes.
Or anything like that? People have been doing this? Or?
Yes, I really wanted to show you one video, but I guess I wouldn't have time to show you. So, for example, in a recent study, a group has created a little kind of swimmer, or it can be adapted to a swimmer, coated with a magnetic ink. And so it is not self-propelled, but we can control is displacement in the body using a magnetic field. So I think this application is very promising. But yesterday, looking at some presentation about self-propeller machine, I was thinking about how we can apply it in a hydrogel to make it more biocompatible, for example, and to have specific functionality, so a drug carrier and so on. But since it's a complicated field, I think that I just have to think more about how to do it. It's not obvious.
Okay, thank you. More questions, Hartmut.
Just to follow up this point, I think there's Lucio Isa from the ETH in Zurich. They have combined microgels and Janus particles. And so they can breathe and also self-propel. It's very nice. Composition of, say, complex microswimmers. So it's an important question. No?
Thanks, Mathilde. Very nice presentation. I have one question, more specific one, regarding your first system that you showed with the pluronic and polyacrylic acid. My understanding is that it does not crystallize because of electrical repulsion. Is that the case?
It doesn't crystallize.
Because of electrical repulsion. Because I'm just going to ask if that's the case, if you have reduced pH to protonate the acrylic acid part.
Yes, it is protonated.
What pH are you?
It's below 4.
Okay, but still have some percentage of charge. So maybe if you decrease a bit further, wouldn't you be able to form the gel? Because in this case, you can get a system that is responsive to pH in a range which is interesting.
Yes, yes. We didn't test it.
Okay.
Yeah, it's right.
Thanks for this very nice talk. I believe that all these big changes in shape, etc., they are really associated with some supramolecular rearrangement. Have you investigated the ultra-sax region to see if you have any change in the overall shape at that point?
No, we haven't done it. We were interested to see, to use ultra-sax. No, I'm going to say a mistake. But no, we haven't done it from now. We were more focused on getting a system that works, and now we are going to understand a little bit more what is going to happen. And I think I would be very, it will be very helpful to have collaborators in that field, because I'm not such an expert in sax. So we do basics measurement, but not such. Yes.
USP will have a very nice instrument there that can help you there.
Okay, thanks.
More questions for Mathilde? If not, let's thank again.
Thank you.